Ladies and gentlemen, it's our honor to take part in this workshop. Our presentation is real entities through actual causal relationships. The cases of the transgenic and the gene clock-out-clock-in method. I am hard of hearing. My English pronunciation is not so clear, smooth, and fluent. So I will read the contents of the slide. In the stage of question and answer, Jonathon is responsible for hearing and answering your questions. Many thanks. My AI system of my tablet can immediately transcribe your questions and answers so that I can understand what you are saying. We have finished a draft of our paper. If you have read it, then you are welcome to ask questions related to the contents of the paper. You are welcome to email your comments to us after this workshop. Hricko was invited to visit my department in 2007. He also visited the laboratory of Bose-Einstein condensation in the Department of Physics at my university. Since then, I was influenced by him and become an advocate of experimental realism. But I did not work on this issue until I met Jonathon in 2014. I published a paper to defend his approach and expand the number of criteria of reality to include the individuation, observation, counting, in addition to manipulation. This is my paper. Jonathon and I propose experimental individuation and the experimental determination of number per unit as criteria of reality in previous work. The two papers, the second paper published in 2023, this paper defines entity realism beyond manipulation because experimental determination of number per unit is not experimental manipulation. We are practice-based pluralists about the criteria of reality. This time, we will propose a new criterion of reality that combines manipulation with causal inference. Entity realists typically relies on either manipulation or causation to demonstrate the reality of entities. We develop an approach that combines both. We argue that the reality of entities and of the causal relationship they stand in can be demonstrated by satisfying the following criteria: manipulating a specific cause to make an actual difference. The criteria involve manipulating entities as a specific cause under certain conditions and an understanding of causation in terms of actual difference making and causal specificity. We argue that experiments using the method of transgenesis, gene clock-in and gene clock-out, satisfy the criteria, thus demonstrating the existence of a particular gene that is taken to be the specific cause of some phenotypic feature. There are three questions about the manipulating a specific cause to make an actual difference. What is the precise meaning of MSC? Why does MSC count as a criterion of reality? How do scientists demonstrate the reality of transgenes in their experiments based on the MSC criteria? The criteria involve understanding causation, in terms of the concepts of actual difference making and the current specificity that has been developed by Woodward and others and used by others. Woodward summarizes his interventionist conception of causation as follows: X cause Y if and only if there are background circumstances B such that if some single intervention that change the value of X and no other variables or where were to occur in B, then Y or the probability distribution of Y will change. Woodward continue by offering interpretations of key concepts in this definition. First, variables X and Y are properties, quantities, which can be assigned different values. Second, an intervention on X with respect to Y as an idealized experimental manipulation of X, which causes a change in Y that is of such a character that any change in Y occurs only through this change in X and not in any other way. To explain the causation in classical genetics, Waters explicitly introduced the distinction between potential difference making and the actual difference making. He argued that there is a genuine distinction between identifying a mere cause, that is the potential difference maker, and identifying the cause that makes the actual difference, that is the actual difference maker. The phrase to identify one gene as the cause of a particular phenotypic effect implies the notion of causal specificity that is expressed in the following paragraph. DNA is a specific difference maker in the sense that different change in the sequence of nucleotide in DNA would change in the sequence in RNA molecules in many different and very specific ways. RNA polymerase does not have this specificity. Intervening on RNA polymerase might slow down or stop synthesis of a broad class of RNA molecules, but it is not the cause that many different kinds of intervention on RNA polymerase would change the DNA sequence in RNA molecules in many different and very specific ways. However, the functional difference between the sequence of DNA and RNA polymerase only shows that the DNA is the causally specific potential difference maker. Waters asserts DNA is the actual difference maker because there are many such differences in DNA do actually exist, and these differences actually explain the specific difference among RNA molecules, indicates that the DNA is the causally specific actual difference maker with respect to the population of RNA molecules. First, synthesized in eukaryotic cells. The vector basis of Waters' assertion that the DNA is a specific actual difference maker need experimental endorsement. The techniques of gene modifications offer strong support because scientists actually intervene on the sequence of DNA and make actual phenotypic difference in engineering. in organisms in experiments on gene modification. In those experiments on gene modification, experimental organisms display phenotypic difference when scientists transfer or remove a sequence of DNA into or from the organism. There are three methods in gene modification. All of us is transgenesis. It is to introduce a gene to the genome of an organism that does not have the gene in a natural state, observe whether or not a new phenotype is expressed. The transferring process is written. The second is the method of gene clock-in. It's generous gene to a targeted locus of a genome in an organism or substitute an original gene in a genetic locus with another new one. Observe whether or not a new phenotype is expressed. The third is gene clock-out. Targeted gene is an organism is deleted from or made inactive in the genome of the organism, comparing the phenotypic difference between the clock-out organism and the normal individual. The fourth method is transgenesis. This method uses the following procedures. The first is scientists use registration enzyme to cut segments of DNA at the correct size and then use DNA ligands to combine different segments of DNA. The second is to introduce a foreign gene into a new genome. Scientists use technique of recombination DNA to link its generals of DNA segment to a vector. Those vectors are typically viruses, bacterial phages, or plasmids. The vector transports its generous DNA segment to the target cell. An example is cut DNA from plasmid and combined with DNA segments that encode antibiotic resistance to produce E. coli cells with antibiotic resistance. Concerning the case of gene modification, prima facie, scientists manipulate a gene to modify phenotypic features of an organism so as to demonstrate the existence of the gene. It seems the Hacking manipulation criterion of a complete answer. However, one may double that one can demonstrate the reality of transfer genes simply through the experimental manipulation of the gene because we first need to demonstrate the reality of the causal relationship between a transgene and the phenotypic features that the gene is purported responsible for. To demonstrate this, it seems that we need a lot of theoretical knowledge and our assumptions about genetic mechanisms. For example, the encoding of DNA, the transcription of genetic codes from DNA to RNA, the translation of codes from RNA to synthesize proteins, and so on. We do not need theoretical knowledge assumptions if we apply what was notion of actual difference maker and causal specificity. Applies these notions, we need only consider whether or not a sequence of DNA actually makes a specific difference on phenotypic features of modified organism. Although Woodward and Wallace's notion of actual difference maker and causal specificity help us select a specific vector of the cause of phenotypic change from multiple vectors. These notions are not conceived to demonstrate that the cause is the real entity. In the case of gene modification, moreover, what is intervening and what is manipulated are different entities. We need a formulation of causation more complex than many traditional definitions of causation to fit with the case in genetic engineering. Here we propose the formulation of specific actual difference makers. X is the specific actual cause of an actual difference G in Y if Y is intervening by manipulating X and the actual difference G between intervening Y and another intervening Y is made by this intervention. The terms actual contrasts with not only potential but also theoretical, hypothetical, model, and idealized, referring to the actual application of causation in laboratories. The meaning of the term specific is the same as that specific in Woodward's and Wallace's notion of causal specificity. A specific actual cause would always occur in a context of actual intervention or manipulation. A specific actual cause is the real cause. If an entity plays the role of a specific actual cause in a causal process, then the entity real exists. If an entity is manipulated to make a specific actual difference, then the entity plays the role of specific actual cause. As a consequence, we drag the MSC criteria manipulating a specific cause to make an actual difference, demonstrate the reality of the entity that plays the role of the specific cause. Applying this criterion of real causation and the MSC criterion of reality to the case of transgenic experiments, we will send transfer the gene as X, the experimental organism as Y, and the specific feature that the transgene is purportedly responsible for as Z. In summarize, there is an actual difference between the experimental intervening cells and the other natural non-intervening cells of E. coli. This transfer gene makes the actual difference. Therefore, the transfer gene is the specific actual cause of the antibiotic resistance. Such an experiment satisfies the MSC criterion of reality, manipulating a transfer gene as a specific cause to make an actual difference. Therefore, the transfer gene exists. There are several limitations of transgenesis. So scientists develop the methods of gene crack in and crack out. But due to the limitation of time, I have to speak the two methods. This is a diagram of gene crack in. This is a diagram for gene crack out. Now, we have to make a comparison with other views. First, Hawkins' view. He said, we are completely convinced of the reality of electrons when we regularly set out to build the over enough succeed in building new kinds of devices that use various well understood current properties of electrons to interfere in other more hypothetical parts of nature. However, what are the so-called current properties? Which notion of causation do they depend on? Hawking does not provide the answer to these questions, nor does he connect manipulation with causation. Our view relies on manipulation, but manipulation alone cannot demonstrate the reality of unobservable entities. What we need, in addition to manipulation, is a way of understanding causation in terms of current specificity and actual difference making. Manipulation is what scientists do. This understanding of causation is what justifies the inference to reality of unobservable entities. As a result, this understanding of causation is doing most of the work to support entity realism. Now, we make a comparison with the colorist view. The colorist view is proposed first by Wright and developed further by Clark, Suarez, and Egg. Clark and Suarez use inference to the most probable cause to infer from an effect to the reality of its cause. Egg use inference to the best explanation that satisfy certain conditions, non-redundance, material inference, and empirical adequacy to provide current warrants for claims about the reality of unobservable entities. Our view is the currentist approach to entity realism has two open questions. How do we understand causation? And what specifically, what are the causal relations that support reason about entities? We suggest understanding causation based on what was interventionist notion and Waters' notions of actual difference making and causal specificity, as we have argued about. The colorist answers also raise a problem concerning the distinction between causal and theoretical explanations. We suggest finding such causal relationship from experimental cases per se to avoid this problem, as we have argued about. Our position on entity realism. According to our position on entity realism, we should provide evidence for the reality of an entity based on actual practice, including experiment, observation, counting, specific causal inference, without depending on theories, models, hypotheses. We can solve this problem. How do we go beyond manipulation and remains the basic position of entity reason without slipping toward standard scientific realism? We believe that it is appropriate to suspend judgment about the reality of entities if there is no evidence from actual practice. Thank you for your attention.